Assalamualaikum, and thank you so much for still be with us. So we now go to our third speaker, which is Associate Professor Dr. Suhaila Abdul Muid. So I think everybody knows Associate Professor Dr. Suhaila Abdul Muid is actually Associate Professor of Biochemistry and now actually Head of Department of Biochemistry, Faculty of Medicine at UITM. So welcome Dr. Suhaila to the session. So now I would like to invite AP Dr. Suhaila to deliver her talk entitled Fundamental Methodology. And just a housekeeping, we can actually post our question at the Q&A tab. All right, the floor is yours.
Okay, thank you, Dr. Rosfaiza, for a very kind introduction. Assalamualaikum and a very good afternoon to all participants and organizer. It is already lunchtime, so I hope you all can bear with me and stay until the end of this talk. So this workshop is continue with this topic, fundamental methodology. So I will deliver this topic in general, but if you would like to have a detailed explanation about the topic, feel free to contact me or to ask me question after this session. Okay, so for understanding better on the fundamental methodology, so we go a bit of introductions on fundamental research. So fundamental research, also known as basic research and pure research, it generates new knowledge and technologies to deal with unresolved problems. Meaning, if you have a research question in the area of your interest, so if there is a gap in that research area, so perhaps that can be a fundamental research on its own. Okay, so this fundamental research contributes towards the enhancements of knowledge, and in the long term run, it is the basis for many commercial products and applied research. So the research should focus on basically accumulation of theories, fundamental structures, and fundamental processes. So a successful proposal write-up basically is not about who researchers are, it's not about what researchers need, and it's not about researchers' background and history. So it is about what researchers do, what need researchers serve, and researchers' vision and future. So it's not really about the reputations of the researcher. So it is really about the idea of the research. Okay, so according to the Liddy and Ombrod, research proposal is actually lays out the problems for research and describes exactly how the research will be conducted and outlines in precise detail the resources. So basically, this will be covered in the methodology where with this the researcher will use to achieve the desired result. Okay, so again, I would like to highlight about the characteristic in the quality of research proposal. First, of course, you need to have a informative title, and again, according to Prof. Musalma, you need to have a very convincing and persuasive executive summary, clear problem statement and objective, scientific background and rationale, good selection of research method, ethical considerations for clinical research and animal study, and of course, realistic budget and schedule according to the what is the budget that's been provided by the grant provider. So we focus on the good selections of research methods. Okay, so the importance of methodology sections, basically it tells your research committee how you are going to plan or tackle your research problems. So it will provide your work plan and describe the activities necessary for the completions of your project. So many proposals are turned down due to unsound methodology, especially when the methods are not aligned and unrealistic amount of work that they want to carry out. So so objectives must be cleared and with detailed description, and the applicants must explain how they plan to carry out and measure each objective. And again, the most important is be realistic in designing your research design according to the objectives. Okay, so basically applicants must provide answers to the following questions. So what is the experimental model or design, and what activities need to meet the objectives? What are the start and the finish date for the research activities? So again, the durations, you need to look at the durations of the research. Usually it's being provided by the grant provider, whether they want to you to apply the grants for 2 years or 3 years. So you need to be clear on that when you write your research proposal, and who has responsibility for completing each activity, and what facilities and equipment will be required to conduct the activities. So usually we will choose the facilities and equipment that is available in our locations. Otherwise, if we don't have that facilities and equipment in our locations, you might need to find it in another locations, and you may need a co-researcher from that site. Okay. So basically, this cartoon tries to tell you that the selections of methods cannot just based on your gut, on your or your instinct. So it must have basis and align with objective. Okay, like this one, maybe the supervisor tells the students, maybe yes, you can pick that method, but as long as it wasn't based on your gut. Okay. Okay, so in the methodology also need to include flow charts or experimental design, research activities, milestones, and as well as Gantt chart. Okay, so in medical research, so basic research is under primary research, along with the clinical research and epidemiological research. So I think for clinical research and epidemiological research, it will be covered by AP Dr. Zaleha. So for me, I will cover on this basic research, and I will highlight these methods under this basic research, which is the in vitro and animal study. Okay, so the procedure and experimental designs in the basic and fundamental research can be specified and implemented, um, meaning in the basic and fundamental research, you can actually control the environment. And for example, you can also define the populations, you can actually define number of groups, case numbers, treatments, and dosage where they can be exactly specified. Okay, so that is about basic and fundamental research. So for fundamental methodology in vitro study, so the selections on cell culture model or methods must be based on objectives. For example, if you would like to have a research on the model of diabetic cell culture model, so you need to really design your methodology and find the suitable cell culture model that is really suit your desired research design, which is a diabetic cell culture model. And of course the methods must be based on the objective. And if the cell culture model is already in existence, you need to put the reference in the methodology. If not, how it is superior. So maybe you can explain that your methodology in the proposal is superior to what is already being described in the previous publication. So it is going to be an advantage on your side. And then methods, again, I would like to highlight that methods must be aligned with objective. So this is the example of the fundamental research proposal. So this is the title. I think it has been explained in details by Prohabinder. So again, the title and the objective must be synchronized. So the objectives is best to be put as a general objective and the specific objectives. Okay, so after you have defined your objective, so what will you need to do next? Okay, so you need to plan to carry out, and you need to somehow put your plan to carry out and measure each of the objective. First, you read the objective, and then you define what activities and experiments needed to meet the objective. For example, just now, okay, if we look at the objective, I would like to determine the protein, I mean the effects of oxidized HDL and adiponectin on the formation, on the mineralizations for the first objective. And then I would like to measure the protein and gene expressions of osteoblastic markers in these two cell lines. And then I would like also to determine the effects of oxidized HDL and adiponectin on the secretions of the pro-inflammatory cytokines. And I also would like to study the possible pathway that may involve in the uh, in the uh, in the effects of the oxidized HDL. So in these objectives, I already list down the target biomarkers of interest that I want to measure. So with these informations, so basically I can plan and I can list down what are the activities and experiment that I need to do to meet the objective. And then what are the starting and the finishing dates for the each activities, and who has responsibility for completing each activity, because we as the PI, we may not have that expertise in that specific method. So we need to find the research members that has expertise in that area. Okay, so I think this is going to be further explained by Dr. Jamal tomorrow, and what facilities and equipment will be required to conduct the activities. Okay, like I mentioned just now, preferably if you can choose the facilities or equipment that is available at your location. But otherwise, you may need to find a co-researcher from the other location, or you can outsource it to the supplier, or something like that. Okay, and then okay, so in the methodology sections, so the tips is, it is good if you can have before you describe your methodology in the methodology sections of your proposal, it's good if you have this overview of this experimental design. So this is like to explain the flow of your research activities. So usually, because this is based on the comment that I received from the previous FRGS applications, they mentioned that it is good if you can actually provide this overview of the experimental design, so that the reviewer can really see or feel very clear when when they read your methodology, and they know the flow of your research work. Okay, so for example, like this one, I will tell them what are what are the types of cells that are going to use. Okay, and then what is the treatment that I want to give to the cell, something like that. And then how I determine the various concentrations of the treatment to the cells. So and then once I achieve the optimal concentrations, then I will need to and then I will need to do the to incubate further for measurements of the my biomarkers of interest. Okay, so basically this overview of the experimental design can also be explained in the flow chart. Okay. Okay, so after that, then you can lay out your methods. So this is how the example, the methodology write out in a research proposal. So I took this method from my own FIGS proposal. Okay, so basically I will tell them what is the cell culture that I want to use, and then the incubations of acetylcysteine and adiponectin in the cells, and then what is the methods that I'm going to use to measure this myelinization and calcification assay. And then since I want to measure the gene and protein expressions of these parameters, so I need to put what are the methods of the protein and gene expression that I want to use. So in here, for gene expression, I put here, I choose quantigene plex assay, and for protein, I choose ELISA. And also it is good to have the list of your parameters or your biomarkers of target. So this is the example from my proposal. And as well as since I would like to measure the pathway analysis, so I also list down the pathway or transcription factors that I want to measure in my study. And as well as you need to also explain what are the statistical analysis that you're going to use in your analysis later on. Okay. All right. So this is actually just to tell you about the common methods on protein expression, especially for the newbies kan, for the new lecturers who is planning to apply a proposal. So basically, they don't have any idea what methods that need to choose, especially if you have already have a target protein or target gene. So what method that you want to use? So basically, this is a very basic method to measure your protein expressions. You can use ELISA, enzyme link immunosorbent assay. And if you want to measure the protein on the cell surface of your cell, you can use flow cytometer. So if you have a tissue sample, you can use immunohistochemistry. And as well as Western blot is the same principle as the ELISA. As long as you have the target gene, then you can conduct this assay. Otherwise, if you want to screen your protein, then you need to use the other method such as SCMS. It is a more advanced method. Okay, but I think for FMRG, I think these are the methods that I think suitable to propose in your proposal. Okay, so about the common methods on gene expression. Um, so this is you can choose quantitative real-time PCR. This is, I think, the cheapest method to measure on gene expression. So you can measure gene expression on tissue sample, on cells, on fresh blood. But remember, you can run this quantitative real-time PCR if you already have your target biomarkers, like what I want to measure in my proposal, just like I mentioned just now. Okay, but if you want to screen, then you need to use the other methods. Okay, this is only if you already have your target gene. Okay, so these are also the methods that can be used to measure gene expression. We call it as a gene plex. So just now, real-time PCR can measure one gene at one time, but this one is more advanced method where you can measure more than one gene in one go. So if you have more budgets that been provided by your grants provider, so you can proceed with this method. Otherwise, you can choose real time PCR, whichever. I mean, you need to choose the method that can suit your grant budgets. Okay. Okay, this is actually, there are many more methods that to measure the protein and genes expressions of your biomarkers. So I just name a few, but you can also refer here. Yes, especially here to metabolomics. This is more advanced method where if you want to screen your protein, then you can use this method, LCMS, and as well as this mass spectrophotometry, but we don't have this. I'm not sure, but some of these we have it in our faculty in IMB, but otherwise you can actually outsource it, or you can actually collaborate with the other researcher from the other university or institutions. So again, in the research methodology, you need to have a flow chart. So in the flow chart, you need to clearly show the flow of the research activities, and again, it must be reflect the project objective. And it is very important for you to provide the flow chart. Okay, so this is the example of the flow chart from the in vitro study. Again, I take it from my own proposal. Okay, for example, you can actually draw the flow of your research activities, yeah, until data analysis and documentation. For example, here I put the name of the cell culture that I want to use, and then I want to do the optimizations to obtain the optimal dosage that are going to proceed with my experiment, and then what are my treatment set. So I have this set of cells with treatment, and I have and then you need to lay out, or you need to actually define your control. So you need to have the negative control, and if you have vehicle, then you need to have your vehicle controls. So that is very important, and the reviewer we look at that, yeah. Meaning if you already have that, then you have a proper research design, experimental design. And then you can put here what are the parameters that you want to measure, and then the method that you want to use. For example, here I put real time quantitative PCR for gene, and ELISA for protein, and I list down the biomarkers that I want to measure in a table form. Okay, so this is the example of the flow chart. Okay, so for in vivo animal study, so in brief, you must select a suitable animal model, or specific species according to your research topic or research scope, and again, according to your research objective. And you need to mention in the methodology that you're going to apply for animal ethics approval. This is very crucial, because all animal and also human experiment need to undergo animal ethics approval first before you can conduct the experiment, or even before, if you can start the experiment. And then you need also to determine the sample size determination. So if good, it is good if you can actually include the calculation of the sample size. And then um and then of course you need to lay out about the experimental designs, meaning you need to actually define your treatment groups, and what are your control groups, and as well as you need to include the collections of your sample. Okay, how are you going to collect your sample, and is your sample involve blood, tissue, cells, and also the method of the sample collection. So it need to be properly put in your methodology. And then the parameter measurement methods, and as well as statistical analysis. And then after that, again, don't forget to put the experimental design, and as well as the flow chart. But for animal study, I think it's more to the experimental design. So this is the example of the experimental design. So I took from one of the publications as cited here. So basically, this is the time of the experiment. So you can properly put in the experimental design the grouping of your animal, and then the durations of the animals that need to be induced, for example here with high fat, and then what are the treatment that they will receive here, the dosage, and here vehicle is the control, and it is and then the durations of the treatment until the end of the experiment. So the experimental design usually must be self explanation, yeah, must be self explained. All right. So this is the example how you can write your research activities. So you can list down all the research activities from the beginning, and you must have the starting date and the end date. It depends to the grant provider. So some of them just want you to list the research activities, and some of them want you to define the starting and the end date. So just follow the guidelines of the grants. Okay, so usually it can start with ordering of consumables and method optimizations, and then of course the optimizations of the other methods of experiment, and then you can start your actual experiment, and then how much is the durations, yeah, and then until you can actually list down all the research activities until publications and report writing. Okay, so this is how you can write your milestone. So milestone is actually about the completions of the date of completions of each of your research activity. So it's maybe overlap with the research activities, but in the milestone, you need to actually really put what are the important research activities, and what are the date of completion for each of that, yeah, so that especially for the FIGS grant, they will use this to monitor the progress of your project, yeah. For example, for the first quarter, maybe they expect that you have finished the first milestone, yeah, and depend first and second milestone, but of course it's also according to the date that you have stated in the proposal. Okay, again, I took this from my own proposal, and then some of the grants provider wants you to actually list down the the equipment that you want to use, and the facilities that you want to use, and the locations of the equipment and the facilities. Like here, all the facilities that I choose here is actually available in IMMB. Yeah, so it's quite straightforward. Otherwise, like I mentioned before, maybe you need to find co-researcher at the other locations. Okay. Okay, then in the research methodology, usually you need to m- put the research outcomes. So usually the research outcomes, you must put the expected findings. So what are the expected finding that is that apa expected finding from your research proposal. Usually the expected finding must can apa must be benefit lah, must be beneficial to the maybe patient care, to the management of the patients, to the communities, improve quality of life. So usually the expected findings must be leading towards that, yeah. And then of course you can put there your planning for the publication. This one is very important because all grants provider wants to have publications as the research output. So you can actually put the impact factor of the journal, and you can always mention the journal name, and it now we always aim for the Q1 and Q2 paper, so it's like a bonus point for you kan, if you can actually mention that in your proposal kan. And then so this publication that showing that you have a plan, so it is very important to put the apa publications and number of publications in your proposal. And also it depends, some of the grant provider need you to put this human capital, yeah. And again, please refer to the guideline what they need in terms of this talent of the human capital. Maybe for example in the FIGS grant, For 2 years, they want to have one master student as an output, yeah. And for 3 years, they want to have a one PhD. Or there are also small grants, they don't require um, postgraduate student, but enough with just one, if you just train an RA, yeah. So it depends, so please read the guidelines. And you need to also to elaborate the potential applications, and elaborate what are the new knowledge is actually covered by your research proposal, and what are the impact on the society, economy, and the nations. Okay. I think this is the last slide. Okay. So again, I would like to stress on the point that the proposal, like what Prohabida mentioned this morning, must be synced from start to the end, meaning must be synced from the title until the end of your research proposal, yeah, until to the budget, yeah. So the research reviews must support your problem statement, and then your research question, hypothesis, and your method must related to the objective and must answering your objective. And then again, method must reflect member contribution, and flowchart summarize the method. Outcome must follow the grants criteria, and again, budget must be reflected to the methodology. Yes. So again, please look the guideline of the proposal, and you must be realistic, yeah, in terms of your objectives and the budget. All right. Okay, so I think that's the end of my presentation. Okay, so I give back this session to AP Dr. Rosfaiza. So I welcome for any question and answer. Thank you.
Thank you so much, AP Dr. Ila, for very nice and very informative talk. Uh, Also include what are the methods that we actually can use when we write a proposal. Okay, if
Yeah.
you have any question, you can put at Q&A. So I have, while we wait for other question, I just
Okay.
wonder, AP Dr. Ila, we always have a first time when we write a proposal again. Instead of writing, we always has our first time. So I just
Mm-mm.
wonder because I mean, like there are lots of methodology, and I mean, when somebody write first time, right? Sometimes we do have some idea, but we don't have any really knowledge about what are the method that is suitable for us to do the to put it in the proposal. So I just wonder from your experience, is there any tips? I mean, like sometimes we want to approach the co-researcher, but we cannot approach them by empty methods, right? We need to put something on the proposal.
Mhm.
Do you
Yeah.
have any tips as such we can start off with some method at least when we first time write the proposal.
Okay, I mean, according to my
Mm.
experience, okay, thank you for the question, AP Dr. Ros Faiza. It's very important. According to my experience, because this is from my from my postgraduate studies lah. Okay, so basically at that time, my project is on the inflammatory cell culture model. So I really don't have any idea what is the method all about. So of course, before I want to approach my supervisor, or before I want to approach the other co-researcher of my project, first, of course, we have to go back to the literature search publications. So, so at that time, before I discuss with them, so I look at the literature. What are the established method on the at that on the inflammatory cell culture at that time, and then and is the method is actually suitable for my research design. Yeah. So basically, that's what I did last time. So I look at the literature search, and again, if you have someone in your lab that can help you, you can always refer to that, especially if that person have experience with that method before that. Yeah. So maybe you can get their advice. Kan maybe the the research design maybe not maybe the same, but maybe you can always discuss, kan, have an idea, and then after that, then you can approach any of your co-researchers, or your or even your supervisor. So that's what I did last time. So, and again, in order to select the method, first you need to look at the research objective. And for the beginner, I think it's good if you can choose the basic method first. But depends lah to the amount the amount of the grant that you receive. So let's say if you receive apa banyak I mean if you receive a high amount of money, then you can straight away go to the advanced method. But of course you need to find a collaborator that is actually expert on that methods, yeah. Otherwise you can choose you can actually choose what is actually available at your lab or at your location. For example, you can visit IMB kan. Let's say if you are really a new researcher kan, you don't have any idea what is the method available for your research. So even if before if you want to write the proposal, you can come and visit IMB, or you can ask advice from any of the IMB member what are the methods available. So with that, then you can go back and you can write a proposal based on what are the methods available. I mean that is tips lah for the beginner, I think. Later on, then you can develop more advanced method. Then you can find more. I mean, you can find maybe if not available in the IMB, then you can find at the other places. So that one is also good because it can actually build the collaborations among the researchers kan, especially with different universities kan. So it's quite a KPI nowadays kan. Okay, so that's my answer to Dr. Ijah question.
Thank
Okay.
you so much, Dr. Ila. So, Dr. Ila, here just one question actually
Mm-mm.
from Dr. Nohafiza Razali. So, I read it for you, yeah. So,
Mm-mm.
Assalamualaikum, Dr. Suhaila. When we put the potential papers for the publication output, should we include which Q1 or 2, 3, 4 impact factor and etc.?
Yes, good question on that. Yes, preferably yes. So you can actually put the title of the I mean the name of the journal, but of course it is highly recommended that you put the journal from the Q1 and the Q2. Yeah, from the Q2 journal. Especially it will give you advantage kan for the reviewer when they read your proposal, then they will know that, oh okay, this basically this study have direction, and this researcher wants to publish their publications in Q1 and Q2 journals kan. So it's I think it's good if you can include that, yeah.
But following that question,
Mm-mm.
Dr. Saila,
Yeah, yeah.
I wonder, let's say if you did not manage to get to Q1 or Q2, the ones that you promised, um, and you managed to Q3, probably Q4.
Mm-mm.
How would that? I mean, would that sort of tarnish a little bit of your reputations to the provider? Probably, I'm
I
not sure.
I think in this current FIGS, yes, because basically when they do the evaluation, usually the output or the publication output that you are planning to apa to produce, especially reflect your research proposal. So meaning indirectly you need to produce a research proposal that is actually suit for the Q1 and Q2 publications. I think that's what the current FIGS apa nama applications all about. But for the other grants, I think should be okay. For example, like internal grants that provided by the UITM. Yes, let's say if you cannot publish it in the Q1, Q2 journal, I think as long as you publish it in the index journal, I think it should be okay. So far I don't encounter any problem with that.
Okay, thank
So they
you.
do accept. They do accept. No worry. FIGS, yes. I think I'm also quite worried with that because like
But
what I
as long
mentioned.
as they are setara lah, right? Let's say if you
Yes.
say that you're going to publish in Q2, but it's not that particular journal that you
Yes.
mentioned earlier, but
uh,
it's still considered as sort of similar level, right?
yes, that's true. That's true. But the best, you try your best. Tapi kalau tak dapat juga, then
What
it's
to do?
so scope. Yeah.
True,
So,
true.
index publication will do. Scopus index. apa namanya, my side. Yeah, especially for the internal grant. Of course, with the amount of budget, we
Yeah.
cannot really put a very high-fi punya experimental design inside the proposal. So I think they do accept the apa the index publication. Yeah. Okay.
Thank you, Suhaila.
Welcome, welcome. Welcome, Dr. Hafiza.
Okay, happy to Tyler. There's no question yet. Again, maybe I can ask another question. So again, we just yeah. The other thing is a very simple question. I think everybody think about this as well. Like literature review, sometimes we
Mhm.
go through proposal who write so much things on the methodology. It's so long and so detailed.
Mhm.
So again, do you have any tips in terms of the amount that you need to put? I mean, the details of the methodology in your proposal. Any tips
Okay.
on that?
Usually in the literature review, we don't put methodology in details. Usually you can always mention in the methodology sections, meaning you can put reference, you can cite the reference on the on I mean you can put reference on what of the method that you have been choose from the publications. But I think in the literature review it should be focused on the on the apa research question, on the problem statement, introductions of your research topic. So that is the priority lah for the literature review. Unless if your research proposal is about method optimization. That
Hmm.
one you can actually put the literature review in details about the method of optimization. Other than that, you can especially you can actually mention in the literature review. This is based on my experience lah. In the final paragraph, I will write in the one paragraph basically my objective, and in my objective usually I put what are the methods that I want to use, but of course I will explain in details in the research methodology. So maybe I will put what are the principles. I mean, it depends. Sometimes you don't have to mention in details, but if you want to mention in details, so you can put maybe the principle, or maybe if you want to show that that is the current method, meaning that is the most current method available at that time, you can put the reference, the current reference. So it will give impression to the reviewer that actually you choose a current method that is available. Maybe if you put in 2021, so they know, oh, this is the method is established, is a very new method in 2021. So it will give you um, apa namanya, advantage jugalah to the reviewer, that there's a high possibility for them to consider your research grant. Yes.
Okay, I think there's no more question. Thank you so much, AP Dr. Suhaila, for very nice talk. Thank you for example and all that. I think we are very clear on how we can actually write fundamental methodology.
Okay.
Thank you so much for your time, AP Dr. Ila,
Thank you
on
for
behalf of the
inviting me.
yeah, thank you so much.
Okay.
So
So,
we end the session. So I'm going to share again the QR code for the CPD points. So if anyone can actually have not scanned, they can actually scan this. All right. Okay. So with that, I end the session. I think we are going to continue again at um, 2 p.m. Is that right? Yep, it's actually at 2 p.m. For a session with AP Dr. Zaliha on a talk on clinical methodology. So we'll be here again at 2 p.m. All right. So thank you so much. Thank you again, Dr. AP Dr. Suhaila. And until we meet
most
again at 2
welcome.
p.m. today. Okay.
Thank
Assalamualaikum.
you.
Thank you.
Thank you,
Thank you
Fiza.
so
Thank
much.
you. Thank
Bye.
you to
Thank
all.
you.
Have a nice lunch.
You too.
Bye.